Hello guys, we are back with evolution and biodiversity. So let's cover the topics that we didn't cover previously. I hope you're all doing well. Okay, so we have three domains of life. We have bacteria, archaea, and eukarya. Alright? Bacteria and archaea are prokaryotes, okay, and there are several differences between prokaryotes and eukaryotes. An important thing to just remind you about the cell wall of bacteria. So bacteria have a cell wall, and they have peptidoglycan, so protein and sugar, and this is only present in bacteria. So eukaryote animal cells don't have peptidoglycans in their cell membrane. So antibiotic medicine that targets peptidoglycans will only affect bacteria, right? So that's one way to design antibiotics, just targeting features that are present in bacteria, for example, and are not present in human cells. Okay. So there are two main ways to collect energy from nature. One is autotrophy, where the organisms make their own food. So a plant, for example, obtains its energy from sunlight, directly from sunlight, so they're primary producer, and from environmental atmospheric CO2, carbon dioxide. Okay, so this is photoautotrophs, and plants and algae are autotrophs. And we have also the case of the marine deep sea vents, where there is volcanic activity and a lot of inorganic chemicals are released to the environment, and there are some bacteria that evolved to be able to use this and the carbon dioxide to make their metabolism and their structure. So instead of collecting light, they will be collecting inorganic chemicals. Okay, so just to remind you that we are carbon-based life forms, and here this band here, it's a very good Swedish band. So if you want to look at them, it's carbon-based life forms. You can find them on YouTube. And really, all of our molecules have a carbon skeleton. So complex biological molecules almost always consist of carbon atoms that are bonded with other elements like oxygen, hydrogen, nitrogen, phosphorus, or sulfur. Okay? So that's the backbone of life. There are theories about the possibility of having a silica-based life forms in other planets, maybe, but nothing is confirmed. It's just a possibility. Then we have heterotrophs. So, for example, a giraffe, it cannot consume energy directly from sunlight, so it will eat the primary producers, it will eat the plants and obtain the energy collected by the plants, right? Then we have secondary consumers like the lions that are not herbivorous, so they need to eat meat. So they will eat, in this case, a giraffe, for example, that ate a plant, and the plant took energy from the sun. Okay, so it's important for you to have these concepts in mind. Okay, so let's get to bacteria. We have these important cyanobacteria, which are very old, in between 3.8 billion years or 2.3 billion years ago. And these are structures called stromatolites, and basically it's layers of deposited bacteria, cyanobacteria, and you can find these in Australia. So there is like huge areas of stromatolites, and what these are cyanobacteria colonies. Okay, and yeah, again, in between 3.8 billion years ago or 2.5 billion years ago, this photosynthetic bacteria that have chlorophyll started to collect CO2 from the atmosphere, and also they had the sunlight, right? And back then, 2.5 billion years ago, there wasn't any oxygen on the planet. So as they did photosynthesis, they started to generate oxygen. And this changed dramatically the atmospheric environment that we see in our planet. So this was the origin of oxygen-rich atmosphere. Okay, and that changed everything. Also the origin of chloroplasts. So this will be a chloroplast, each of these single unit of cyanobacteria that was then later engulfed by a bigger bacteria or cell. So that's the endosymbiotic theory part of it. Okay, so we get into protists now. Basically everything that doesn't fit into plant or fungi or animal, you throw it into category protists. It can be called a kingdom, but there is a lot of debate, and also again phylogenetics are constantly changing, so new hypotheses are being tested, and instead of kingdom there is a division in supergroups. Okay, so there is this one supergroup here, these that will be the plants, here we have animals and fungi, and here another supergroup of protists. So protists can vary in size, they can be microscopic or macroscopic, also they can be unicellular or multicellular, as we will see, and they reproduce both sexually and asexually, and also autotrophs or heterotrophs. So we have carnivorous protists. So last class we went into land plants, and now let's go back, so let's come down the tree. Okay. So in the supergroup Archaeoplastida we have several groups. We have rhodophytes, the red algae that we see here. They have pigments. They have different pigments, but phycobilins and carotenes give them this red coloration. They have chlorophyll A and chlorophyll D. Then we go to chlorophytes. So we have these examples. Here we have Spirogyra, which has spirally arranged chloroplasts. And here we have chlorophyll A and B. Charophytes, which are the closest to land plants, as we can see here, right? Also have chlorophyll A and B. There are unicellular and multicellular, and they have motile flagellated swimming cells. So yes, and then we go into land plants, so the embryophytes, and they also have chlorophyll A and B. So yes, this is just a quick overview on archaeoplastids. Let's move on to the supergroup SAR, straminopiles, alveolates, and rhizaria. So here we have several groups. The straminopiles include diatoms, which we saw maybe from the first class that we had under the microscope. And they lack flagella, so they don't have this swimming flagella like sperm has, or these other archaeoplastids. They make their cell wall is made up of silica, so glass, and they make these very intricate structures, and is a very diverse group. And also, okay, so it's a diverse group, and they produce around 20% of the oxygen that we have. And the chloroplast has 4 membranes. So on primary endosymbiosis, we have a chloroplast with 2 membranes, and the fact that it has 4 membranes indicates that it happened as a secondary event of endosymbiosis. We'll not get into details on that. Brown algae. So maybe you know the kelp forests. You have marine forests of brown algae called kelp, and you have them on the California coast and also in Europe and all around the world, basically. And they have this pigment, fucosantine, that makes them olive green to brown. Oomycetes were previously organized under fungi because they have many similarities to fungi, but actually they belong to the straminophiles. Okay. In alveolates, we have the dinoflagellates, which are responsible for this bioluminescence that you see sometimes on the beach. In my hometown, back in Portugal, during the summer we have this kind of phenomenon of bioluminescence in the sea. It's green, not blue. Also responsible for red tides, which are toxic and have many impacts on fisheries. And just for information, this is the dinoflagellate, this structure. And it has 2 tecas. Okay. The ciliates are covered in cilia, so they're like these hair-like structures around the body, and they swim using that, so they swim much faster than most of other protists. And they have 2 nuclei. They have a micronucleus, which is only used for reproduction, and they have a macronucleus, which is used for cell regulation. They have a mouth pore, so they are carnivorous, they feed. And under alveolates, we have apicomplexa, sporozoans, which includes Toxoplasma gondii, which is responsible for toxoplasmosis, which is a disease that is transferred by cats and rats. And interestingly, these parasites, Toxoplasma gondii, can get into a mouse brain and also a human brain, but in the cats it cannot cross the blood-brain barrier, so it can't get into the brain of the cats, but it can get into the brain of rats and humans. And there are some research that suggests that these parasites on rats makes the rats attracted to the smell of cat pee, and so the assumption is that there was coevolution between these parasites and the cats to help them attracting prey, basically. Okay. So Rhizaria, it's another supergroup in which we have foraminiferans. They make this spiral, like almost snail-like structures with calcium carbonate, and they have this thread-like pseudopodia. So pseudo means pseudo, right? And podia means leg. So almost a leg. And we find this in the coast of England in Seven Sisters Cliff. So all of this cliff is made of foraminiferans, and this cliff that you see here previously was at the bottom of the sea. So remember, these are marine organisms that sedimented, okay, and now are above ground by tectonics and just all of the movement of plates. Radiolaria, we have this, I think, very cool structures, and they have also pseudopodia that are called axopodia, but instead of calcium carbonate they have a silica skeleton like the diatoms made of glass. And also all of this is sedimenting at the sea bottom, like deep sea. Okay, So here we have another supergroup, the excavates, which includes euglenids, parabasalids, and diplomonads. Euglenids have both chloroplasts, and they also have a mouth. So they are mixotrophs. Mixotrophs means that they are both heterotrophs and autotrophs. So they can get their energy from sunlight, but also they are predators. They have this flagella, okay, that helps them to swim, and they have an eye spot. That's a photoreceptor called stigma. That's here in the mouth entry, basically. So they can detect shadow and light. Parabasalids, these guys here. The disease trichomoniasis, the most commonly sexually transmitted disease, is caused by Trichomonas vaginalis. It's a parabasalid, and they're excavates. Okay, and the diplomonads are actually two cells that are bound together. So there, and so there is one nuclei and another nuclei, and they have 4 flagella each. So you end up having this structure, and they lack mitochondria and Golgi apparatuses. They have mitosomes that substitute the mitochondria, and they are mostly parasitic. Okay, so this Giardia lamblia also can cause disease in humans. Okay, so now we go to the supergroup Uniconta that includes the amoebozoans and the opisthocons. So let's look at the amoebozoans first. We have amoeboids, which include amoeba. So here we have amoebas, just a single cell with pseudopodia, so it can move by dragging these pseudopodia. And then we have slime molds, which we looked at already, Dictyostelium, right? And they also have an amoeboid stage, but then when they fuse, it's just a cytoplasm with a lot of nuclei, because nuclei undergo mitosis, right? Without cell division. Okay, so let's get into opisthocons. Here we have heterotrophs only. So we have animals, fungi, and quinoflagellates. This is a quinoflagellate. Okay. It's a unicellular opisthocont, and what we see is that this organism, it's very, very similar to the choanocytes that exist on the chambers of marine sponges, of porifera. So here we have strong evidence for evolution, cellular morphology evidence. It's really very, very similar. And so these choanocytes have the function of bringing seawater into the sponge, creating a water flow. So this is a marine sponge. And they are animals. So you have to remember that the sponge is an animal. Okay, under opisthocons we have mushrooms, and yeah, so we have Eumycota, true mushrooms, and metazoa, animals. Okay? Okay, so fungi are mainly decompositors, so they decompose dead matter and basically bring death back into life. So all of the dead materials are reintroduced into the cycle by decomposition, and fungi are the main responsible for this cycle. They do not digest the food inside like animals do. They release enzymes to the extracellular environment, and then they absorb the nutrients already dissolved, so they are sapotrophic, where they absorb the food. and some of them are parasitic. They produce spores and reproduce both sexually and asexually. So I'll not get into much detail in fungi, but just here some important things to have in mind. So fungi make mycorrhiza with plants, which basically is the plant root as a connection with the hypha, the mushroom, the fungi root, and this will help the plant to absorb nutrients, and also it connects one plant with another plant. So in some cases you have, if you go to a forest, it's most likely that all of the plants are connected by a network of underground fungi, and there are some really cool ideas about this being a sort of internet where they exchange information, be it electrical or chemical information on the environment. There are good books about that, and here there is a secondary literature. So we have BBC News. I'll give you the presentation so you can check this link. And also the largest living thing on Earth is a fungus that basically occupies miles and miles of land as a single organism with a single genome. So like sort of avatar-like. Yep. And here we have this video of Paul Stamets, which is a renowned mycologist. So if you want to also take a look at that. Here I have these reindeers, because in Siberia the tribes in Siberia, they use Amanita muscaria, which is a hallucinogenic mushroom. They use it during the winter time, and also the reindeers, the wild reindeers, they eat this mushroom, and so they use it in ritualistic, in rituals and in religious practices. And so there are some people that support the idea that the Christmas story to be very much inspired by that. So we have the colors, the red and white that come from Amanita. This one, that's Amanita. And maybe if the tribe is hallucinating, maybe they can see the reindeers flying in the sky. And also the main mycorrhiza connection they have is with pine trees. So we have the pine Christmas tree, right? So we have these two main groups, the most advanced fungi. We have the Basidium mycota, which include puffballs, bracket fungi, jelly fungi, and the fungi that you eat normally. And then we have ascomycotes, include truffles and morels and coral mushroom. Okay. So let's get into animals. We are still in opisthokonts, right? We have a conoflagellate ancestor, and we have a sponge here, most basal to all of the other animals, and closest to this conoflagellate. And a few important things to remind. Protostomes, the blastopore will develop into the mouth, and in the deuterostomes, the blastopore will develop into the anus. So let's take a look in that. And okay, so here we have the only phyla that are deuterostomes, so that the anus develops first, which are chordates, that includes fish, reptiles, mammals, and echinoderms, which includes sea urchins, sea star, crinoids, so on. So this is the only invertebrate that has deuterostome development, the echinoderms. Okay. Alright, so we have protostomes then, that the blastopore will develop into the mouth first, and this includes Spiralia, so all of the organisms that during development, so after the egg is fertilized by the sperm, right? it will become a zygote, diploid zygote, and it will need to divide, so it will start dividing, right? 2 cells, and then 4 cells. Oops, sorry. Well, you get the idea, right? So 4 cells. And spiralia, they have this this way that the cells divide and they move as a spiral, as it's depicted here. So those are spiralians, and they include the annelids, mollusks, lophophorates, flatworms, and rotifers. Ecdysozoans, which include arthropods, insects, arachnids, and nematodes. This is a nematode. It's a microscopic, almost microscopic worm, flatworm, sorry, roundworm. They have radial cleavage, not spiral cleavage. Okay, so they don't have this spiral movement. There is a question on your McGraw post quiz that asks about protostomes having spiral cleavage. So let's assume that all protostomes are spiralians, which is not true. Okay, so ecdysozoans here don't have spiral cleavage, but for the McGraw quiz, assume that they have. Okay, I already wrote them to correct that. Okay, so let's move into arthropods. Basically, arthropods are the most diverse group of organisms in Earth. At least that's documented, that we know the species exist, because of course we are always finding new species, right? But we have more insects than we have plants or fungi or virus or bacteria. So that's impressive. And here we have, we saw the sow bug, right? And probably now we'll start to see some ticks if we go out. So be careful with that. And so on. We have lobster, that's also an arthropod. Spiders are also arthropods. Scorpions, those are arachnids. So we have a lot of diversity, and we're always finding new species. Okay, so now let's get into the Protostomes. These include chordates. So here we have fish, zebrafish, that's a chordate, right? It has bones. And here we have a sea urchin, which is a model organism for developmental biology and embryology. And here we can see that they don't have spiral cleavage, right? There's like this radial symmetry always. This is sea urchin. And chordate development is a bit more complex, we'll not get into that, but we can see here 2 cells and then it starts dividing. Okay, so that's all. Now let's get into some instructions for your virtual lab. So all of the progress is saved, so you don't have to do it in one sit. I suggest that you do it focused and like reading everything and making it and looking at the videos. So if you need to take a break, you can take it and maybe do it later or do it one day later. It's up to you. But instead of like forcing yourself to do it in 1 hour, maybe you'll not be as focused as you would be if you do it, let's say, every morning or something like that. It's actually quite simple and fun exercises. So I made another video to show you how it works, and just at some point you reach DNA comparison in these novel labs, and so we have these numbers. We have these numbers 1, 5, 10, so on, that will mark the position of the nucleotide that we are looking at. So it should be simple. If it says there is a adenine in position 2, then you look here. This is position 2 here. You look here and look at what animals have A at position 2. It's only this Eoseti. I'm not sure if that's an animal. So that's just an example of how biological research is done. Here I have some data from my work. These are not nucleotides; these are amino acids. So actually, what I'm looking here is at the phylogeny of this protein, which is an enzyme that catalyzes several reactions. So, for example, to make dopamine, this enzyme is required, and also to make serotonin and some other tryptamines. So here I'm comparing the differences of this protein across phyla. So we see here we have a human and snake. So here we have vertebrates, we have snake and frog and fish, and here we have the sea anemone, which is what I work with, which are cnidarians. So it makes sense that this protein, this tree that I made, appears so far away from vertebrates, right? Okay, and then if you want to send me also your tree that you made after the Nova Labs, so by each mission you will unlock a piece of this, and then you can make your tree shapes. Okay, so send me this tree also together with your documents that you submit, and I will take 2 points if you don't, because that's my way to see if you are looking at the lectures or not. Okay? Take care.